sir. Okay, sir.
Okay, so ma'am, shall we start the event?
Yeah.
Okay,
Definitely.
okay. Thank you, ma'am. So, welcome to the invited lecture and interactive session series organized by departments of chemistry and departments of physics, Durgapur Government College, under the DBT Star College scheme. So, as a coordinator of the scheme, we are very grateful to Sir for giving his valuable time for our students and faculty members. So, to start with the inaugural event, I would like to now request our principal, Sir, to please share the welcome notes. Sir, please.
Yeah. Thank you, ma'am. Am I audible?
Yes, sir, you are perfectly audible.
Okay. Thank you. Good evening to you all. On behalf of Durgapur Government College family, I'd like to express my deep sense of gratitude and heartfelt thanks to our resource person, Honorable Dr. Padmaja Prasad Mishra. Despite his busy schedule, he has agreed to give his vast experience or knowledge in presence of our the students, faculty members also. My sincere congratulations to the coordinator, DBT Star College Scheme, and head of the department of both the department, both physics and chemistry, as well as the other faculty members of both the department, and departmental coordinator of under the DBT Star College Scheme, my beloved student, research scholars. So, my heartiest welcome, sir. So, though it is in online mode, but I would like to invite you in the physical mode in near future. And without wasting valuable time, I hope this session will be most interactive and enriching session. Students, please interact with the eminent resource person. So, with these few words, I wish a grand success of this interactive seminar invited talk. Thank you so much.
Yeah, thank you, sir, for the welcome note. I would now like to request our head of the Department of Chemistry, Dr. Snigdha Chandra, to please introduce our resource person. Ma'am, please.
Thank you, Dr. Acharji. Good evening, respected Principal Sir, Professor Mishra, respected coordinators, DBT Star College Scheme, respected colleagues, research scholars, and my dear students. A very warm welcome to you all. Today we have our respected Principal Sir with us. So I would like to take this opportunity to thank our Principal Sir for his continuous encouragement and support for all the academic endeavors that are going on here at our college under his tutelage. Today, we are delighted and as well as honored to have with us Professor Padmaja Prasad Mishra, Associate Professor, Chemical Science Division of Shaha Institute of Nuclear Physics. Professor Mishra will talk on visualizing DNA in action, a multidisciplinary research methodology approach. This lecture series is a joint initiative of DBT Star College Scheme of Durgapur Government College and being jointly organized by the Physics Department and the Department of Chemistry. I would like to thank Professor Mishra for being with us today and for taking out time for Durgapur Government College. Now, allow me to put forward a few words about Professor Mishra. He has asked me to keep it as brief as possible. So, following his instruction, let us have a glimpse of his academic profile. Professor Mishra has obtained his postgraduate degree from Baharampur University, Odisha in the year of 2000 and has received his PhD award in 2005 from IIT Bombay. His field of research was biophysical chemistry and spectroscopy. He obtained postdoctoral degree from the University of Pennsylvania, USA. He worked there from 2005 to 2007 and then got another postdoc degree from Pennsylvania State University, USA, where he worked from 2007 to 2011. After that, he came back to India and joined the Saha Institute of Nuclear Physics in 2011. His research interests are DNA repairing, biophysics, single molecule imaging, and many other fields related to biophysical chemistry. He has published many papers in reputed international journals with high impact factors. Dr. Mishra has a very sophisticated laboratory there in SINP, and and his team have done many Foster resonance energy transfer studies of different systems. You can get those papers on SINP website or as well as on the personal website of Professor Misra. Today, he will talk about multidisciplinary research approach. So, students of different science disciplines will be benefited from it. And I believe our students have developed a certain level of understanding about DNA research from the Science Setu lecture series. I would request all of you, my students, to interact with Professor Misra after the completion of the session. And we welcome again you, Professor Misra, on behalf of the Department of Chemistry, Durgapur Government College. And I request sir to please start the technical session. Thank you, sir. Now over to Professor Misra.
Thank you so much, ma'am. Thank you so much for the introduction. I asked you to be brief, but it like you said many things, many good things about me. Let me justify it first. Um, Very good evening, all of you. Thank you, Dr. Palit, for extending the invitation. Signa ma'am for coordinating with me. Like many times, I've not even able to send all the documents she has asked in time. But I think the day is here. Whatever mistake has done is okay. It's time to discuss something exciting, something new. I believe there are many undergrad and master students there. And during the discussion, I got to know that there will be students from physics, there will be students from chemistry, there are students from biology. So I thought of choosing a subject which is common to all. What else could be a better topic than talking about DNA? The best chemical compound, which has a wonderful physical property, and the most important members of any biological society. So let's talk about DNA. I'll just start sharing the slides, and if the slides are visible to all of you, I can go ahead and start presenting.
Yes, sir.
Yeah, good. So, as I said, we'll be talking about DNA, and mostly it's going to be a multidisciplinary research methodology approach so that students can get involved. I'll not talk more about the research work. We can talk about that thing if someone is interested, someone wants to visit the lab, want to look into the system or look and get interact with the other research scholars. We can do it later. Let's make this topic very basic so that students can understand, they can develop their interest. Uh, well, it's DNA. Why DNA? So the things that I'm going to talk about is what DNA, why DNA, and how we can look into DNA. These are the three things I'm going to cover. What is DNA? Most of us know it. It's deoxyribonucleic acid, the most abundant chemical substance present in nucleus in all the living organisms, the cells of all the living organisms. If I just give a number, 90% of the human DNA is even same from person to person. This is the chemical compound which controls all the chemical changes that takes place in the cell. This also controls what kind of cell it is going to form. It's whether it's going to form a muscle cell, it's going to form a blood cell, it's going to form a nerve cell. This is being controlled by DNA. And what kind of organism also it will produce. Whether it will produce a giraffe, it will produce a chimpanzee, or it will produce a human, or it will produce an insect or a plant. That will be also that is also governed by DNA. So that's why I said a subject which can be understood by many aspects, many disciplines, across the disciplines. And when we are talking about DNA, as I said, 90% of the human DNA are like unique, but that 1% less, the rest 1%, that makes us different. I am different from you. My son is different from me. My parents, I am different from my parents. All that is because of that 1% DNA. Even if we talk about twins, their DNAs are different. They are not similar. So what DNA does? It affects the appearance. It controls the behavior. Am I okay?
Yes, sir. You are okay, sir.
Yeah. So it controls the behavior. It controls the health. Why I'm telling it control the health? Because of DNA, there are many diseases also related to DNA. And to control those diseases, to treat those diseases, we need to treat the DNA as well. So the DNA basically is, as we all know, it's inherited from our parents. So we share the traits from both our parents. The siblings share the traits also, and we eventually pass the traits to the children. So, like like the way you use our fingerprint for our identification, 1901 DNA printing, DNA fingerprinting is also been utilized for many reasons. Identify a particular person, to identify a particular organism, and it has been implemented in. We all know that from nucleotide based RNA, RNA structures look compounds.
Sir, excuse me, sir. Your voice is actually not clearly audible. Yeah, sir, you are not audible, sir. So you are still not audible, sir.
Is it better, ma'am? Now?
Yes, sir. Now you are audible, sir.
Okay.
Thank
Better.
you.
Thank you, ma'am. So, coming back to the nucleobases, the adenine makes 2 hydrogen bonds with thymine, and guanine makes 3 hydrogen bonds with cytosine. That's why it is called as complementary base pairing. And yes, and these bases are basically joined by a backbone that is called as a sugar phosphate backbone. This sugar phosphate backbone, there is a sugar here, which is a ribonuclease, and connected with a phosphate. So this is the one strand of this DNA, and there are 2 similar strands of this DNA. So as I said, adenine makes 2 hydrogen bonds with thymine, cytosine makes 3 hydrogen bonds with guanine, and this is how it makes, and this is how the structure of DNA are made. So in this structure, initially, if you look for the primary structure, it just looked like a ladder that we use at our home. 2 strands, which the 2 backbones, which are basically the phosphate and glucose backbone, the ribose backbone, and there are the nucleobases, they are hydrogen bonded in between. And eventually, just to get an optimized structure, they get twisted like this, which is called as a helical structure. This is a very typical structure that DNA makes, that is called the DNA helix. They make a helical structure, and eventually they get compacted and make higher order structures. So the DNA, like as I said, now it makes different kind of higher order structures. It makes helix, stem loop, pseudoknot, A form, B form, Z form, stem pseudonodes, histones, holiday junctions. There are other form of oligonucleotides which are called as RNAs, and many other structures. For And all these structures comes while they execute different kind of biological function inside the cell. So to understand the DNA, we need to understand the function of these different structures. We need to understand how these functions execute, the role it is associated with, and this is very important. To execute all these biological roles, as I said, it attend different kind of conformation. I'm just giving some conformations here, but other than that also, they attend different kind of conformation. If there is a different platform some other day, I would love to explain many other things about DNA also. But to a chemist, to a physics, or to a fundamental biologist, this is what it does. I was telling one more thing, like this is how DNA behaves inside the cell. But other than that also, this DNA, other than executing the biological function it is associated with, for many reasons, and to execute those biological roles, it attends different structures, different conformations. I have said those things already. Many of the time, not because only the biological roles, because of many extracellular and intracellular activities, this DNA experience different kind of damages, different kind of other conformational changes. And if there is a damage in the cell, suppose there is a UV radiation fall on the DNA, they induce different kind of damages. For other cellular regions also, salt concentration, that many of the cellular activities that induces damage inside the cell. And if those damages are not repaired or not taken care of in proper way, they end up of having different diseases. And it's very important to tackle those kind of damages also. And that needs the understanding of the function of the DNA. And this is how the methodologies come. We use different tools to understand those things. One of the common form that DNA experience inside the cell is something called a bubble-like structure. This is what I'll giving example. So this is a like a bubble, which also form because of many biological activity like DNA replication, transcription, DNA repair, recombination, and many other places also, and because of UV radiation and other activities. And these bubbles are formed because of thermal fluctuations, because of change in environmental pH, because of the characteristics activity of the DNA that is associated with its biological function, and many other things. So it's important to understand DNA. If I'm a chemist, I would look from a window of a chemist to understand the function. If I'm a physicist, I would look from the window of a physicist to look into the function. And when chemistry and physics combine together, they can give better solution to the biologist to understand the biological function. So how do we utilize different tools to understand, be it is DNA, be it is protein, be it is any other chemical system, be any of the research, we utilize different kind of tools. I'll just describe in brief some of the common tools that we use in the research. One of the common tool is spectroscopy. We use different kind of spectroscopic techniques: UV visible, IR, fluorescence, NMR, mass, EPR, ESR, Raman, and most of these spectroscopic techniques are there in our course structure. If you are in BSc, you are in MSc, you must be going through all these spectroscopic techniques. So I'm not explaining them in details. We use spectroscopy. What we get from there, we get to know about the properties, how it is behaving without seeing it. But it is believed that seeing is believing. So we can use microscopic tools to visualize them. There are different kind of microscopic tools also, like microscope, electron microscope, fluorescence microscope, scanning probe microscope, and there are many. There are advanced microscope like atomic force microscope. There is super resolution microscope, and many other things. We are not discussing that thing, but the microscopes are there where you can visualize, and the more you visualize, the more you understand them. There are light scattering techniques like dynamic light scattering, static light scattering, relay light scattering, which give you information about size, which gives you information about the distribution and all these things. X-ray diffraction, we get the crystal structure. Computational or theoretical calculation, we get the energy optimization, the best structure that is possible. We use different kind of chromatographic techniques. If we are in organic chemistry, we are doing some kind of synthesizing some compounds. We also use many kind of chromatographic techniques. Similarly, it is used in biological field also, like paper chromatography, thin layer chromatography, column chromatography, gas, FPLC, HPLC, and many more. There are centrifugation techniques which are used based on the mass, the size, to separate molecules, separate systems. Electrophoresis system, where you separate systems using the charge. So what I did is I roughly described many tools that usually we use in a chemistry laboratory, in a physics laboratory, or in a biology laboratory to understand systems. So whatever the tools I described here, if you look carefully, all the tools that are here, they are kind of similar tools, where we take a large number of molecules together, be it is a spectroscopic techniques, be it a microscopic techniques, or anything. We take a lot of molecules together and we analyze it. When we take a lot of molecules together and analyze it, we lose many informations. I'll come to that. So, for example, we are just using a very basic absorption spectrum, like I've mentioned it here. One absorption spectrum, which looks a very broad spectrum to us, it consists of a combination of many sub-peaks, but we can't. So, and each peak is characteristics to some of the nature. But when we look into the absorption spectrum, it looked like a single peak, and we thought, or we think, we consider them as a single substance, but it's not actually the case. So because of we consider a lot, so in all these techniques, we consider a lot of molecules together and analyze them. They're called as ensemble technique. One of the traditional techniques that we have been using, and it will be used for another, like for a long time also, because of their robustness. There are many advantages of using this ensemble approaches, where you consider a lot of molecules together, because they are very robust. You don't have to, you get a very good signal when we use any of these tools. If there is a microscopic observable, like color, solution, color the solution, temperature, and all these things, we can actually continuously measure and continuously monitor also. However, if you're talking about a large number process, let's say we are talking about a reaction. For example, if you take a reaction, if you take a solution of 1 microliter of 1 micromolar solution of any compound, that contains 6 into 10 to power 11 molecules, and we are not sure how these molecules are behaving there. So what we think, whatever information we are getting there are being observed by all the molecules. There, what we are compromising with the actual behavior the molecules are possessing. So these are the disadvantages, because we are average the system. If the system is stochastic, it's very impossible to synchronize it, and we are not following what exactly all the molecules are doing. When we are dealing with a class of 30 students, we cannot say that all the students are good, all the students are bad, because every student is individual, and when we assess them, we have to look for individual students. Similarly, if you want to know more information about a large group of molecules, let's say, we have to look for individual molecules. This is what the advancement is going on nowadays, and many people are working on different advanced techniques like single molecule techniques. I'll just give one example today because it's a research methodology part. I'll just give simple way of explaining how can we also look molecules individually instead of looking taking a lot of molecules together. I'll just whatever I said, I'll just give you a small example. Why I said no, if there are lot molecule, that is the information that we get biased. If you look at the screen, I'm just showing some random matchsticks. Some of them are vertically oriented, and some of them are horizontally oriented. So if I ask you to take the average of the orientation of the molecules, this would look something like this, which none of the sticks are oriented in that way. So the average does not say how the exact orientation of the molecules. But whatever we get from the ensemble methods, mostly we get the average over a number of molecules. That's the reason only I said the averaging would be biased. So many of the time we don't get the exact information that we're looking for. So how to overcome that? As I said, a new techniques is being evolving. It's not there in any of the course that we cover in our BSc, in our MSc, that is called as single molecule measurements. This is a very new tools that, like, nowadays, if you go back, Google into and Google single molecule methods, you can get a lot of informations. But still, it will take some time for them to include in the regular course structure. So what is so single molecule measurements means you look individual molecules. Instead of looking for lot number of molecules, you look individual molecules and see how they are doing. Particularly if you are dealing with systems like DNA, systems like any kind of proteins, cells, nuclear, it's way much better to look into individual system than looking for a lot of molecules. That's why so there are many advantages of looking into systems individually. And I'll just tell you one. There are many single molecule measurements. I'll just cite one single molecule measurements that is been used by many biological methods, many chemistry and physics labs as well. That's why I'll tell these methods here. And I said this is very useful studying complex processes, because so the cartoon that is visualized on the screen is the protein that travels when we do our, I'm not sure if it is visible. Yeah, when we contrast our muscle, this is the protein, it moves here. So, so we can monitor all these complex biological processes using single molecule tools. So one of the advantages of using single molecule tool is if there is a distribution, if there is a population distribution, be it static, be it dynamic, that we can monitor. If you're monitoring something at very complex chemical processes, A is going to B, is going to C, is going to D, and so on, you can monitor those things. And you can detect single biomolecules under biological relevant conditions, so it's easier. Works at very low concentration, so it's easier to do. It's not very difficult. But the thing is, I'm not sure how many of you have heard about single molecule tools before, but can we actually catch hold of a single molecule? Yes. If I'm telling that thing, definitely we can do that. I'll tell you how we'll do that. But yes, we can do that. One of the ways, using fluorescence methods. There are other ways also. So fluorescence methods, I'll just tell what fluorescence is. I guess most of us are familiar with Jablonski diagram. If there is a molecule, we excite it, it goes to the excited state, which is called as absorption, and we get absorption peak. And there is a non-radiative process, it can come back to the ground state. But if there is a radiative process, we call that, where the molecules come back to the ground state, it is called as fluorescence. So absorption spectrum is always at lower wavelength. Fluorescence spectrum is always at the higher wavelength, because there is some loss of energy before the molecules come back to the ground state. So this is fluorescence. We know what fluorescence is. There are phosphorescence. We are not discussing about phosphorescence, but fluorescence at least no. So during this fluorescence, that happens a very interesting phenomena. Snigdha ma'am was briefly telling about that thing, which is called as Foster resonance energy transfer, or commonly it's called as fluorescence energy transfer. Fluorescence resonance energy transfer. What is that? Suppose we have a molecule. We have a fluorophore and we excite it. So fluorophore means any molecule that fluoros. That is fluorophore. Suppose we have a fluorophore here and we excite that fluorescence with a light and we get the fluorescence from there. Suppose there is another fluorescence nearby the first fluorophore. So when you are getting the fluorescence from the excited from this molecule, that means that molecule is at the excited state and coming back to the ground state. Suppose there is another molecule nearby. So there is a possibility that instead of this molecule coming back to the ground state, it will, whatever extra energy it has, it will donate to this molecule, so that it will come to the ground state, not giving fluorescence, and this molecule will start giving fluorescence. But we are not exciting this molecule. So this process, whatever I have explained, is called as fluorescence resonance energy transfer. This fluorescence, so the molecule which donates the energy is called as donor. The molecule which accepts the energy is called as acceptor. Very simple term. And this fluorescence resonance energy transfer can happen if and only if the molecules are nearby. If the donor and acceptor are very close to each other, the efficiency is less. If the donor and acceptor are if the donor and acceptor are very close to each other, the energy transfer is higher. If the donor and acceptor are away from each other, the energy transfer is lower. So that means we can calculate the distance between the donor and acceptor, particularly if the donor and acceptor are within a range of 1 to 10 nanometer even. And any minute changes that is happening within this 10 to 1 nanometer, we can get information about all these things. That's why this process is called as, FRET is used as a spectroscopic ruler to monitor minute changes in the range of 1 to 10 nanometer. Conditions, there are certain conditions the donor and acceptor need to make, but the thing is, the distance is, the efficiency is more if the donor and acceptor are very close to each other. As the distance between donor and acceptor increases, the FRET efficiency decreases. So now coming back, so now I guess we are kind of clear that what FRET is. Now I'm coming back how single molecule FRET works. So for FRET, I am sure it's well understood that we need a donor and we need an acceptor to monitor FRET. So to do single molecule FRET, we have to put the donor and acceptor in a particular place where we can monitor them. For example, let's say we are monitoring the movement. I'm just giving a random example. Suppose we are monitoring the movement of a football player, a soccer player. And if you want to monitor the motion of the conformation of that football player at different instances, what we can do, and we want to use this FRET to monitor that thing, what you can do, we can put the donor and acceptor in both the hands, so that whenever there is a hand movement, we can monitor that thing. We can put the donor and acceptor labeled on one hand and one leg, so that we can monitor the coordination between hand and leg. We can put the donor and acceptor on both the legs as well, so that we can monitor the coordination between both the legs. And to get more information, we have to do all these three things. We have to put at both the hands, we have to put hand, leg, we have to put both the legs, and any other possibilities if there are, so that we get more information. And in this way, we can do the same thing in biology. We have to put a donor and acceptor at two different positions of the molecule that we are interested in. And whenever there is a conformation change, we can monitor that. So once we put the donor and acceptor, we dilute it. As a chemist, if we think of dilution, it's like adding more solvent and diluting it, but there are other ways to do that. That will take time. I'm not going to discuss it here, but if someone is interested, I can explain it sometime later. Then there are certain steps we have to do it. One of the important is excitation using total internal reflection. The physics students, the physics friends and colleagues who are sitting here, probably they understand this in a better way. What is total internal reflection? Suppose we have a system and a light is passing through rarer medium to denser medium or denser medium to rarer medium. And we know that according to Snell's law, it will either bend away from the normal or bend towards the normal. If the refractive index of this medium is greater than, if the refractive index of this medium is greater than this medium, it will move away from the normal. So this is what it will happen. And how much it will move, that depends on this angle. So that's why it is written n1 sine theta 1 is equal to n2 sine theta t. So if you keep increasing this angle for a particular angle, so increasing angle means this light will keep bending. For a particular angle, it may so happen that this light may just come perpendicular to this normal, like this way. If we keep increasing the angle, this process is called as internal reflection. And the angle for which this internal reflection happens is called as critical angle. For example, if you are talking about a glass water interface, the critical angle is about 61 degree. What will happen if you still keep increasing this angle? If we still keep increasing this angle, what will happen? The beam will come, touch the surface, and come back to the same medium again. That is called as total internal reflection. Something interesting happens there at that time. At the point of interaction, some electrical disturbances creates and they propagates parallel to the normal, as you can see it here. These electrical disturbances are called as evanescent wave. I'm repeating it again. These electrical disturbances are called as evanescent wave. What is evanescent wave? Evanescent wave means the wave that is vanishing. The good point is these waves generate at the interaction and they carry the same wavelength as the excitation beam. They travel about 50, 60, 70 nanometer depending upon the power of the light, and they vanish. And that is the advantages of using this TIR, this total internal reflection, the use of this evanescent wave to look molecules at the single molecular level. Because these waves vanish, they can only excite molecules which are present at the surface within a distance of 50 to 60 nanometer. Anything above that or below that, they cannot even excite. That's the reason of using this evanescent wave. And mostly for single molecule FRET, this evanescent wave is being used. So this is one of the criteria for doing single molecule FRET, where you need to have evanescent wave to excite the donor. You can just check. You can just check the generation of the evanescent wave. Take a laser, like there are many key chains available with a laser light. There are pointers available. Anyone can just do it. Take a glass of water, put the pointer in a particular angle. You can see that it is touching, and make the room dark so that you can see this light coming, touching the surface, and coming back, creating a total internal reflection. Well, you cannot see the evanescent wave, but you can see the TIR happening there. So we use this TIR to do the single molecule measurement. So now coming back to the DNA, we have been away from the DNA for a long time. So coming back to the DNA, so how do we monitor the DNA? So the DNA that, as I said, there are two strands of the DNA, and we want to monitor that DNA using single molecule FRET. How do we do that? Put the donor in one strand, ask them to sit. So you make some arrangements like the way we sit in the classroom. We have particular chairs. We go and particularly sit on that particular chair. Like the way you make seat arrangements for the DNA. This is a chemical modified seat arrangement. I am not going in details. So once the seat arrangements for the DNA are there, we can put those DNAs which are there, which are labeled with a donor, so that they can go and sit on the chair. And you can see they are sitting away from each other. Once they sit away from each other, you can get, using a very sensitive camera that is called as a electron multiplying CCD camera. So we get the intensity of those donors. You can see it here at this pen. So each dot that you see are the fluorescence intensity of a single donor, molecule. So one donor, we see it here. Then we have the second, out of the two strands of the DNA, one strand is already sitting there. Once we put the second strand and label the acceptor molecule, as I said, to monitor the FRET, we need to have the donor and acceptor. Once we put the acceptor molecule, they go there. Once they hybridize, the FRET, the fluorescence resonance energy transfer, happen between this donor and acceptor. Energy transfer from donor to acceptor, and we get the fluorescence signal from the acceptor as well. Though we are not accepting the acceptor, still we are getting the acceptor signal because of the donor and acceptor energy transfer. And the intensity, if you look for the individual fluorescence intensity of the donor and the acceptor, the donor intensity goes something like this, as it is shown in the green signal. The acceptor intensity goes something like this, as it is shown in the red signal. So whenever the red and green are coming closer to each other, that means the donor and acceptor are approaching each other. When the donor signals are going away, that means the donor and acceptor are going away from each other. This is how, if any changes that is happening within nanometer range, like as I said, 1 to 10 nanometer gap, we can see all those conformational changes. That's why this single molecule FRET has been widely used. If you just go back, Google single molecule FRET, you can see millions of papers nowadays. A lot of people are working on single molecule FRET. So this is the advantages of using single molecule FRET. So you can get all the information. So how do we monitor those? If you remember those DNA bubble I was telling in the initial slide. So to monitor the function of this DNA bubble, it is easier now. It is, you can put a donor here, we can put a acceptor here, and if there is anything, you can monitor that. And what we have seen, that as the formation of bubble happens, be it is because of the radiative induced bubble, be it is because of any biological function, these two strands, they keep fluctuating like this. They keep looking for the complementary partner by going near to each other. Once they don't find it, they come back, and this process keep going to continue for the next step of their biological function. And you can monitor all these things in details from those states. So you can find out how they are, and what we have found, like this fluctuation, when it is happening, it stays at different stages for different time as well. So we can monitor all these things, how far it is moving in one step, how long it stay in each particular stage, what is the distance between these two states, and all these things you can monitor. So to understand this in particular, we have created different bubbles at the DNA, at different size of bubbles, and we get a clear information that as we keep increasing the size of the bubble, the fluctuation increases. And this is how it facilitates the other biological function that is associated with. You can calculate, as I said, what is the distance, what is the shape of the bubble, this is one example. Like I'm just giving the bubble as an example. We can do the same thing for any biological system like this. And we monitor all these things. And this is very helpful. Just 1 second. This is very helpful to a physicist or to a chemist. Even these systems are very helpful to design different kind of devices, DNA-based devices. I'm not sure how many of you have heard about DNA-based devices. I'm particularly talking about the BSc students, but you can even like the way we use devices. What is a device? Device is something which can consume some energy and do some function. So we can also design different kind of devices using DNA. Like there are many examples. We can make some architectures. Then we can make different kind of nano machines using DNA. We can monitor different kind of conductive nanowires where you can conduct different. We can send different signals with time. We can make DNA-based computers and many other things. So this, to do all these things, you need to understand their functions. So these are some things I said. The utilization of DNA, not doing their original biological function. DNA, I have already discussed, it is utilized for they're famous for their biological function, but other than that also you can utilize all these things, and you can understand those things using this kind of sophisticated techniques like single molecule techniques. There are other techniques also. So this breathing, whatever I said, like the breathing of the DNA, that can be also controlled. If you are really interested in making a device from there, so I'm not discussing that thing. And you can monitor if because the DNA, when it functions biologically, there are a lot of other entities, there are a lot of other parameters, they affect the function of the DNA. We have also seen if there are other parameters, how the DNA breathing happens. That was published in an article in RSC Advance. We have also seen how these breathings affect if it is in a molecular crowder or in a molecular traffic jam. And we have seen that if there is a molecular crowder, other proteins are blocking it, the breathing also get controls. And it starts interacting with other proteins as well. So the point of telling all this thing is you can meticulously understand very minute changes in a very precise way using this kind of sophisticated instruments. So this paper was published by one of the student, Manali Basu, in JPC just last year. There are many other things also we do, like monitoring the DNA repairing. That is one of the primary work that we do in the laboratory. How the DNA damage happen, if there is a DNA damage happen, how the protein look into the damaged site and start repairing the damaged site, and many other things. So this is how, almost coming to the summary part. So what I like to summarize here is, other than using the ensemble methods, well, when we are the ensemble methods give us the clear-cut idea about how things are happening, the basic information. But to know more details about complex systems, we may use other techniques also. The way I gave one example today, that we can use techniques like single molecule methods, we can use super resolution microscopy, we can use AFM, we can use cryo-EM, and many other things are there. But the more we use those things, we can understand their basic functions, which eventually will help, not only help us understanding the basic function of those systems, but also understanding their biological function. Suppose you want to design some drug for a disease. So you have to understand how to tackle those things also. And for that, you need many sophisticated tools. So there are many tools. If you interact with many people, you can get to know more about those things with time. So I thanks the organizers for extending their invitation to me to go there and talk to. I'll be very happy to go there, go to Vardhan Government College, go to Durgapur Government College and interact with the students, the faculty members. I also will be happy if the students come here, share some time with me, share some time with the students, see how instruments, how labs work, how different research environments students work there, what are the things they do, and interact with them. That will be a good thing also. So coming to the last slide of the presentation. So whatever works I saw you today, these are the contribution from all the students who have worked. These two are very recent joinees. Other than that, most of the works are my lab is functioning from 2011. So all the students, like this set of people have completed their PhD and doing jobs or postdocs at different places, and these people are still working with me. There are the fundings I get from DIE, from Science Institute, HTRV, and I thank you all for extending your patience, listening me, and I'll be happy to take any questions. Thank you all.
Yeah, thank you, sir. It was really nice to hear to you. So, if our students have any questions or our faculty members can just type in the chat box, or they can also directly ask sir for the same. So we have also a feedback section where we will be inviting questions also, and we will be also communicating you by email. That will be very nice of you if you can just answer some of the questions.
I'll certainly be happy to do that.
Yes, sir. So it was really nice because it was very much fundamental. Actually, that what we wanted from the aspects of the DVT Star College scheme, because it is to promote the undergraduate training.
Right.
Yeah, and the best thing what you mentioned was the fundamental aspects of Jablonski diagram. This they have in their course actually. Physics as well as chemistry students generally have. And we have in the honors and undergraduate courses, the fluorescence and phosphorescence also. Also the Snell's law, the total internal reflection, these are in the physics courses. So in the compact lecture, there was everything for our students, I would say. The chemistry, the physics and biology. After all, we are talking about the DNA. And the last thing, the best thing I would say, you have just simplified your vast research and very advanced research among the students. So you have explained it like the students can ask, not the researchers. That was the best part of the lecture, and also explaining the quantitative measurements and by using animation. I think students are very much excited for the same. We will be sharing this lecture with our students. As well as we will be sharing the feedback. And as principals are told, it is a humble request to you, sir, to visit our campus because presently we are in the online mode as our institution has been taken by the Election Commission for the DCRC, and it will be over in June. So we will be very happy to invite you and for such an interesting lecture.
I'll be very happy to join you, ma'am.
Yes, sir. Thank you, sir, for giving us time amidst your very busy schedule and so much of research work. So, thank you. I think we can also take some questions in the feedback and send you. So, we can just wind up the session, and thanks to our the whole department. Much more thanks to sir for giving his time with us.
Thank you. Thank you so much, ma'am. Thank you, everyone.
Sir.
Good night.
Sir, and very much welcome.
Thank you so much, sir. The lecture was so illuminating.
Thank you, ma'am.
Yeah, I think it is the first lecture which I saw that every student will be able to understand. Because sometimes we have biology lectures which only botany and zoology students are able to understand. And the chemistry lectures, some students are able to understand from chemistry. But this lecture had everything, what every student will be having the flavor of that. That was the beauty of the lecture, I would say.
Nivedita,
Yes,
may I ask, sir, just
definitely.
fundamental, very fundamental. Sir, I am a geologist. But I was thrilled to hear so many things can be done in modern science using DNA. When you explained in one of your slides, that DNA can be used for performing many functions, which hitherto we did not know, that was not known to us. Now, my basic question is, for treating many complicated diseases these days, we hear about gene therapy, genome sequencing, and through that.
Right.
Now, how to connect that with your type of work? Because genes, we know they are also part of, they are linked very much with the DNA type of thing. So how those two things are linked. That is my
See,
one question.
what is done in gene therapy is like there are many. What is, even if you remember, during the COVID session, there are many vaccines which are developed based on the gene therapy as well.
Yes.
So what is done? You take a small gene and you gene. What is a gene? Gene is a small patch of DNA again. Okay, so a small patch of DNA. You, because DNA has a very unique characteristics of, making that base pairs. So they make base pairs like this, they make the base pairs like this, and leave an avenue here to other molecules to interact. And if you take a small DNA, patch it there, you can stop the function of, let's say there is a virus which is sliding on the DNA, you can stop the function of that virus on that particular DNA. This is one example of how this gene therapy works. I have one of my students have also worked on gene therapy. If another way of doing gene therapy is, let's say there is a mutation for some reason, what you do is you take care of that mutation by uh, changing the mutation again. So there are different aspects of doing this gene therapy, and you can actually monitor those things. You can monitor those things using this plate. Also, one of my students is working on that. We have a paper. If you just go back to my website, or I can send you. In 2021, we have published a paper. In 22, we have published a paper where we have actually used a small piece of DNA to silence a DNA that is creating some kind of disease, mostly for cancer. That is doable. That is doable, and you can actually monitor those things also using the technique that we are using at the laboratory.
But it is quite costly. Means these days, even
Yeah,
it is beyond the capability of common
true.
people.
That is true. Like it's not like there is another method. I'm not sure if you are aware of that thing. There is one development. There is only one development in the entire scientific world that is called as optical tweezer. You can go back and you can Google optical tweezer. That is the only development in the scientific community so far which got two Nobels. Once it got a Nobel in '98, and another it got in 2018. So optical tweezer is something like, making a tweezer, making a forceps that we say, using a light beam to tackle DNAs. So that optical tweezer is expensive thing, but it is used in USA and other countries to treat these damaged DNA, genes. Indian government has not approved it yet, but eventually it will be. But that is an expensive thing, as you say. And reaching this kind of advanced technology to common people, it will take bit more time. Even as I said, no, in my presentation, I said these things are not even included in the coursework yet. It will take some more time. Once it became normal, once it became a common subject, it will be there in the course structure as well. Eventually, like the way we read NMR, the way we read mass, this also will start reading it. And other countries have started using this thing. Our country will start using this thing also, but money is a matter.
As of today, it's quite expensive, and
It's quite expensive. I duly agree with this.
Thank you very much, sir. Thank you.
Yes, sir. Thank you, sir. And I would also like to inform the participants, SR has published in Journal of American Chemical Society, which is actually considered one of the best journals of our chemistry background. So we are very happy to have you, sir, with such an exceptional research background, and we would also like to interact with you in our institution.
Thank you.
Yes,
Thank
sir.
you.
Thank you, sir. And we can just wind up the session now. So we are not actually ending, we are basically connecting, sir.
Connecting.
Yes, sir. Thank you,
Yeah.
sir.
Thank you so much.
Thank you, ma'am.
For to have you on our campus.